Bacterial conjugation was discovered by Liederberg and Tatum when they were studying two auxotrophic strains. The first strain is A minus, and A minus is met minus and biotin minus. So it's deficient for the synthesis of methionine and biotin, but it's threonine positive, leucine positive, and thymine positive. On the other hand, the B minus strain is met plus and biotin plus, but it's mutant for the synthesis of threonine, leucine, and thymine. The fact that both are auxotrophic mutants means that if you try to grow them on minimal media, you will get no colonies with either A or B. However, what Liederberg and Tatum did was they mixed A and B in solution in the same culture. And then when they plated cells, and they had to plate a lot of cells, about 100 million cells, and that tells you that this is a rare event. Nevertheless, when they plated 100 million cells in minimal medium, they started to get colonies. These green discs are colonies, and obviously the progenitor cell, the cell that gave rise to each colony, must be prototrophic, and that means that the genotype of this cell is met plus, biotin plus, threonine plus, leucine plus, and thymine plus. Since it's a prototroph, it can grow on minimal medium. But this, if you want to think about it, this is the progeny of a cross between a met minus, biotin minus, threonine plus, leucine plus, thymine plus strain, mixed with or crossed with a met plus, biotin plus, threonine minus, leucine minus, and thymine minus strains. And it's clear that a type of crossover has occurred where the met plus and biotin plus of the B strain have been recombined with the threonine plus, leucine plus, and thymine plus, the wild type alleles, on the A strain. And this clearly suggested that there is exchange of genetic material when you mix the two strains. Bernard Davis showed that this exchange of genetic material actually depends on bacteria touching or conjugating with each other. And he showed this by using a filtering apparatus, using a very fine filter, which would allow compounds or chemicals in the medium solution to pass through, but would not allow bacteria to touch each other. And when he used such an apparatus to mix the A and B strains, no prototrophic colonies were recovered, implying that the transfer of this genetic material actually requires physical contact between bacterial cells. Now, certain empirical observations about bacterial conjugation made it clear that the ability to conjugate was itself a heritable genetic factor. One aspect is that the transfer of genetic information is asymmetric. So a donor strain can give it can conjugate or create recombinants with a recipient strain, but not the other way around. Okay. Um, the other observation was that strains can lose the ability to be donors. And that clearly implied that there was really something that they possessed that could be lost. And the third factor was, or the third observation was, that strains can regain the ability to donate genetic traits or to create recombinants by association with other donor strains. Now we know that the this heritable factor is something called the fertility factor, and what it actually is a plasmid that the donor strains or donor cells have. So this is episomal DNA distinct from the bacterial chromosome that strains that can donate genetic material possess. And such cells that have the ability to be donors, they are called F plus strains. And recipient strains, recipient cells that do not possess the fertility factor or the F plasmid are called F minus. What the F plasmid does is it allows donor cells to make these long projections called pili, and the singular is a pilus, that attach to recipient cells and draw them closer to the donor cell. And once the bacteria have conjugated, then the F plasmid undergoes a special type of DNA replication called rolling circle replication. And what's special about rolling circle replication is that, unlike regular DNA replication, where a double-stranded DNA molecule gives rise to two double-stranded daughter molecules, in this case, only a single-stranded DNA molecule is produced, and this single-stranded DNA molecule travels through the pilus into the recipient cell, where once it enters the recipient cell, the single-stranded DNA molecule is converted back into a double-stranded molecule, and ultimately, when the transfer is complete, will form the fertility factor or the F plasmid. And if that happens, then this F minus strain would get converted into an F plus strain, because if it has the entire F plasmid, the entire fertility factor, then it would carry all the genes that are required to make the pili and do rolling circle amplification and so on. Now, along with F plus strains, another type of donor strain is known, and those are high frequency recombination strains, or HFR strains. Now, there are a few distinctions or differences between HFR strains and F plus strains. The first is that high frequency recombination strains are so called because they produce recombinants at 1000x the frequency of F plus strains. Recall, in Liederberg and Tatum's experiment, they actually had to plate 100 million cells in order to observe very few prototrophic colonies, suggesting that the chance of creating recombinants is maybe 1 in 10 million or 1 in 50 million. However, HFR strains will produce recombinants at a much higher frequency, 1,000 times more than F plus strains. Another observation is that if you cross an F plus strain with an F minus strain, then it's quite common for an F minus to get converted into an F plus strain, so that the recipient strain gains the heritable ability, gains the F plasmid, and so it becomes a donor strain itself. However, if you cross a high frequency recombination strain, so an HFR strain with an F minus, even though they have much higher frequency of producing recombinants, they will produce very few F plus. So although there is transfer of genetic material happening when you cross an HFR strain with an F minus strain, it's very rarely transferring the genes on the F plasmid or the fertility factor, and it's not transferring those fertility genes that are required to convert an F minus strain into an F plus strain. And as it turns out, all these observations can be explained by the fact that F plus strains look like this, which is they have a chromosome and they have the F plasmid carrying all the fertility genes or the fertility factor. However, an HFR strain looks like this, where the F plasmid is integrated into the bacterial chromosome. Okay, so HFR strains have fertility factors, so they have all the fertility genes required to make pili and do rolling circle amplification and to send DNA across the pilus into recipient cells. However, all these abilities are integrated or are carried on the bacterial chromosome of on the chromosome of the bacterium, rather than in a separate plasmid. Now, in order to understand why HFR strains produce recombinants at 1000x the frequency of F plus strains, however, they rarely donate the fertility factor, we have to understand how the linear transfer of HFR genes take place. And this was discovered by what is called an interrupted mating experiment carried out by Wollman and Jacob. So what Wollman and Jacob did was they carried out a cross between an HFR strain and an F minus strain. So the first thing to note about these two strains is that the F minus carries streptomycin resistance, so it will grow in the presence of the antibiotic streptomycin. However, the HFR strain is sensitive to streptomycin, and the HFR cells are going to die if you treat them with streptomycin. The F minus strain is sensitive to this antibiotic called azithromycin and another antibiotic called ton, and the F minus strain will die. Those cells will not survive if they're treated with the antibiotics. However, the HFR strain is resistant to both those antibiotics. The F minus strain is lac minus, and so this is a carbon source mutant, which means that if you plate this F minus strain in the presence with lactose as the carbon source and no glucose, no other carbon source but lactose, these cells are not going to survive because they are not able to metabolize lactose. And similarly, they're gal minus, and they're not also able to use galactose as a carbon source. However, the HFR strain is both lac plus and gal plus and is able to utilize lactose and galactose as carbon sources. So the way the interrupted mating experiment works is the following. At 0 minutes, you cross. That means you simply mix the two strains. Then you let them do their thing for 10 minutes. And at 10 minutes, you interrupt the mating. And the way you interrupt the mating is you take the bacterial solution and you put it in a kitchen blender and give it a couple of pulses. And what that will do is that you're shaking the solution vigorously, so conjugating bacteria are going to get unconjugated or dissociated or separated. And so whatever transfer of genetic material was ongoing is going to get stopped at that point. And then once you have interrupted the mating, then you plate them in streptomycin. And what the streptomycin will do is it will kill all the HFR cells. So it will kill all the donor cells, leaving only F minus. Only F minus cells. And then you take these plates, and then you can do selection to see what the genotype of these F minus cells is. So you can treat them with azithromycin, the other antibiotic. You can grow them in medium with lactose or galactose and so on, to see whether they are sensitive to azithromycin and the other antibiotic, or whether they're lac minus, gal minus, or lac plus, gal plus, and so on. And so let's think about the azithromycin selection. So at 10 minutes, when you do azithromycin selection, you see about 5% of the cells grow in the presence of azithromycin. Now, these can't be the HFR cells which are resistant to azithromycin, because we have killed them with streptomycin. And so if there are any cells growing, they have to be F minus cells that have become recombinant. And have received azithromycin resistance, the gene for azithromycin resistance, from the HFR strain. Now, you can repeat this procedure, or rather Wollman and Jacob repeated this procedure at 15 minutes. So they interrupted the mating at 15 minutes, they interrupted the mating at 20 minutes, and so on. And at each time after they interrupted the mating, they then assayed how many cells had become recombinant and what genes they were recombinant for. And so we could kind of make a timeline for the transfer of genes where this is 0 minutes, and the azithromycin resistance gene starts to get transferred at around 10 minutes. Whereas the TON resistance gene starts to get transferred just a little bit later than the azithromycin resistance gene at around 12 minutes. And the LAC plus gene starts to get transferred around 17 minutes. And GAL plus starts to get transferred at 25 minutes. And what this suggests is that there is a linear, orderly transfer of genes in a very specific order, and could also imply that the farther apart the genes are in the chromosome, the longer it takes to transfer them from an HFR strain into the F- recipient cell. Another thing worth noting about these data is that azithromycin resistance starts to get transferred around 10 minutes, but by 20 minutes, the transfer of azithromycin resistance becomes highly efficient, so that 90% of the recipient cells are recombinant. So this is very high efficiency. The one that got transferred the first. But if you go to the GAL plus gene, then the efficiency is only about 25%. So the one that gets transferred the last is transferred at a low efficiency. One final observation that Wollman and Jacob made was that if you waited long enough, so about 2 hours or 120 minutes, you could transfer the fertility factor as well. So it's very rare for HFR strains to donate the fertility factor, but it does happen. And what Wollman and Jacob showed was that it takes very, very long for this the fertility factor to get transferred in this linear order of transfer of genes from the bacterial chromosome. And of course, the efficiency is very, very low. Now, putting all this together, we get the following picture of how HFR strains transfer genes. So in blue, we have the chromosome, and this is where all the markers, all the genes of the HFR strain are going to be present. So you're going to have azithromycin resistance gene and the resistance to the other antibiotic, and then LAC plus, GAL plus, and so on. And of course, the F minus has its own chromosome, which has azithromycin sensitivity, sensitivity to the other antibiotic. it's lac minus, gal minus, and so on. Now, the HFR strain has the fertility factor. So the DNA of the F plasmid integrated into the bacterial chromosome. And there are two ends in the F plasmid. This triangle is the origin, sometimes just written as O, and the square is the terminus. And the transfer of the genes, the rolling circle replication, starts at the origin, so that is the end of the chromosome that enters the recipient cell first, and you go around the chromosome until you get to the fertility factor, which is going to be transferred the last. And so you will transfer the azithromycin resistance gene, TON-R, lac plus, gal plus, and so on. And you have to go all the way around the chromosome in order to reach the fertility factor to replicate it using rolling circle replication and transfer it during conjugation. And one thing sort of worth making clear is it's not really possible to draw things to scale, because this chromosome is millions of base pairs, whereas the F plasmid may be tens of thousands. Of base pairs. And so the way the F plasmid is integrated into the chromosome, you really have to replicate the entire chromosome before you start to transfer the fertility factor. And this linear transfer also helps explain the interrupted mating experiments, because if you interrupted the mating early, what's going to happen is you would just transfer the part of the chromosome that carries the azithromycin resistance gene. Whereas if you interrupt the mating at some later time point, you're going to have transferred more of the genes into the recipient cell before that interruption prevents a transmission of subsequent genes.